So it's my pleasure to start the final, well, not the final session, the final session of talks before the poster session at 5. So we started in Africa, we went over to the New World, and we covered that. And so now we're moving back over to Asia. So the next 4 talks will cover Asia. The first one in this group will be Dr. Pradipsinh Rathod from the South Asia isomer. And Pradip.
This one?
No.
This one?
Yes. We need to start. Fantastic. Thank you. Well, thank you all very much, and thank you for inviting me to this amazing meeting today, because I'm watching all the other isomers and realizing how much there is to learn from all of us, from each other, as well as from the questions. So I should start with a disclaimer, because I'm very nervous up here. I am a professor of chemistry, and I'm skipping an organic chemistry class to come here today. So very intimidated by epidemiologists who speak a different language. But we've had to learn a lot, and I think I'd like to show you that we're doing well. And I think I'm learning the value of doing good epidemiology in combination with classical basic science that all of us are trained in. So we work in India, and we call ourselves the Malaria Evolution in South Asia isomer. Let's see if this works. Yep. And I thought I would start with a very quick introduction that may help Jane too, coming up soon. Clearly, malaria is a very big problem in Africa, and we've also heard about some of the pockets in South America where malaria is a continuing problem, and we'll hear more about Papua New Guinea and other such places. But in India, it's a significant problem. So you see that out of 1.25 billion people, there are a large number who are at risk because of where malaria is very common. It's estimated that there are about 2 million to 10 million cases of malaria per year in India. There are about 40,000 to 180,000 deaths. These numbers that I'm telling you are numbers that are now accepted by the Indian government. When we started the isomer, or a few years before that, the official numbers from the government of India were about 40,000 cases and about 1,000 deaths. There were some very controversial studies that have helped set the record closer to the truth. If the number of illness and death doesn't move you, the economists tell us that the economic loss annually from malaria is about $500 million to $1 billion per year. So India has also made a commitment towards elimination. They're hoping to join in to world elimination efforts to go from 2016 to 2030, and they're looking for a commitment of about $1 billion towards malaria elimination in India. I put some of these phrases in blue to kind of make sure that we as an isomer have a small role to play in all this. I think we can continually check whether some of the numbers agree, whether some of the values of questions that are being addressed by everybody who works in India are the same priority. And I think we bring different ways of looking at things, and it's been very productive, and that's I hope is a major message today, that we're doing things well together. So if you want to contrast what goes on in India compared to what goes on in Africa, one of the things people always point out is that all age groups get a lot of malaria. So if you go into a medical ward in India and you look at the spectrum of people who are quite sick and admitted into the hospital, they show all spectrum with an average age of about 30, men and women. There's a lot of severe disease, includes severe malaria as well as organ failures of different kinds. Of course, there's also falciparum and vivax in India. And on top of all this, there are more than a dozen different anopheles mosquito species, and it depends on who you talk to. The major 5 or 6 anopheles species is what is mostly discussed. The control programs are all revolving around that, and I will show you that anything we look at carefully turns out to be more complicated than the official view. And so the question is, what is the reason why some of these things, why do these things exist, and why there is a gradient? So it turns out that Northeast India up here, which is on the other side of Bangladesh, and it is alongside with Myanmar, this region of India has the highest cases of falciparum malaria. About 95% of the people who get malaria are getting falciparum malaria. There's the highest levels of death, highest levels of illness, and so on. And the other area that also gets a lot of malaria, my pointer disappears regularly. Let's see if I can master this. Okay. Is what we call Eastern India. So Eastern India is to the west of Bangladesh. The white spot over here is Bangladesh. So India wraps itself around Bangladesh. So these are sort of the hot spots of malaria, and this is where a lot of people focus their efforts. But it's a big country with a very large denominator. And even low levels of malaria end up being lots of cases. And I'm going to show you examples of that. So our sites are actually set up diagonally, starting from Northeast India, going towards Southwest India, where you get more and more vivax. And I'm sure Jane will tell us more that if you go even further south to places like Chennai, it's even higher levels of vivax. So we all have overarching questions. Those are the burning questions that make us go to run isomer programs. In my case, for those of you who know me from my basic science work, we're interested in drug development, and we're interested in understanding how drugs fail. And over the last 2 decades, we've learned that parasites are really capable of mutating at extraordinary rates. When Mala was telling his story about computers that are able to beat chess players and learn from chess players and get around the problems of whatever it was, the heavenly move, I see something similar going on here. We're getting smarter and better at making drugs and hopefully vaccines soon. But the more we understand the parasite is capable of mutating in extraordinary ways. And when we were showing this in the lab with lab strains, one of the things I would get asked all the time is, does this happen in the real world? And the isomers gave us an opportunity to go take a look. We also proposed, when we wrote the grant a long time ago, that if a parasite has mechanisms to change as a haploid in extraordinary ways and make changes where they're needed and not have collateral damage elsewhere in the genome, and we know how it happens now, it can probably use such tricks to get around other types of evolutionary barriers that the parasite has to go through. And amongst them would be the vector range, because in India, certainly, when you talk to vector biologists, they have very hard views on exactly which vector carries which parasite in which part of India. It's been studied for 100 years over there, and people have memorized all these textbooks. We'll come to that shortly. Similarly, as you develop immunity, the parasite has ways to evade that in terms of alternate ways of invading host cells, cytoadhering, which leads to different types of pathogenesis. And so these are all questions of interest. And even though we start as people who are interested in drugs and drug resistance, we think these are all related questions in a kind of a very global way. Joe Vanetz was going to teach us Broadway songs. I was going to teach him a Bollywood song that he should whistle, which is, it's all about resistance and mutagenesis. And so Brian did a great job of telling us how important that is to understand that. So our projects are divided into epidemiology, vector biology, drug resistance, pathogenesis. Our partners from India are the National Institute of Malaria Research and the Regional Medical Research Center. These are 2 groups that are part of the Indian Council of Medical Research umbrella that does basic science research and advises the Ministry of Health control programs, the National Vector-Borne Disease Control Program. So it's been great working with them. We also work with state hospitals, such as Goa Medical College, that I will talk about, and Assam Medical College, which I will also mention today. And we have others, but in a half-hour talk, I want to make sure you get the big picture, and I'll show you some examples of what we do. My own lab at University of Washington is interested in drug resistance, but we also have fantastic collaborators from the US. Manoj Durai Singh is the project leader now on pathogenesis. Joe Smith works on, from the Center for Infectious Disease Research, works on cytoadhesion and different types of severe malaria, and I'll mention that too. We always ask at the end of 5, 6 years of working on all this, are we making a difference? And I hope to convince you that in the short term, I think we're already making a difference in terms of how the national program looks at how drugs are being recommended for use in India. I think we're getting to the vector control people in terms of how they evaluate, how they should be looking at that, and even decisions the way they're being made in individual hospitals. And these are not strong recommendations. They watch what we see, and we share the data with them, and then they go about looking at their own data in a different way. In the long term, I think we have opportunities to possibly develop science that will help with disease predictions and maybe even better ways to protect people. So an introduction to India. I told you that we're interested diagonally from the upper right hand of India down to the lower left. We started all of our work in Goa. So Goa, right here, we call Southwest India. It was just based on what we perceived as the sufficient number of patients, sufficient types of different types of malaria, and the right environment in which to learn how to do field work. And the most challenging parts are up here in the northeast. So these parts of India have a per capita income of about $500 a year. These are not very rich places. Goa is probably the richest state in India. It hovers around $8,000, $9,000 per year. And so you have this huge difference, not only in the amounts of malaria, but also the way it's managed, handled, the way the hospitals are run, the kinds of patients who show up for treatment, and the attention that they get. So we're capturing a little bit of all of that by looking across different parts of India. And this map here might capture a little bit about the magnitude of India. I think sometimes we forget that the size of India from one end to the other spans distances that often go over many countries in Asia itself, in Europe, or even parts of Africa, and so on. So given the challenges of doing certain operations and things related to what isomers have to do, we have stuck with India, and we find that we can do a lot of different type of science there. So today I'm going to focus primarily on Goa Medical College and Hospital and the work that we've done there, because that's where we started in our second year, and the National Institute of Malaria Research field site, which is located in the same city, essentially. And that's where we do our vector biology with Dr. Ashwani Kumar. And those two things together have worked very well for us, and that's where we learn how to do field work. And then we also have additional sites across India, but I'm going to end up talking about the Northeast, where I'll talk about the Indian government's setup called the Regional Medical Research Center, which is responsible for understanding health issues in all of the Northeast. That covers about 7 to 8 different states on the other side of Bangladesh. And of course, in terms of overall malaria questions in the world, I have been brought up to believe that most malaria drug resistance begins here in Southeast Asia, and it somehow goes across India and then disseminates into Africa. And all the records show that even in India, you first see drug resistance appearing in the Northeast. This is true about chloroquine, pyrimethamine, sulfadoxine, and so on. But the perception is that it all starts in Southeast Asia. I've always wondered how much it has to do with Nick White. He happens to be there, and he's looking more carefully. He's got great trainees, and they're asking the right questions, and they find things there. And so we'll see what happens in the next isomer, but I want to be another Nick White. I'm going to look here in Northeast India. I'm hoping to convince you that there are good reasons to think that a lot of nasty things might be going on within Northeast India. And we should keep our minds open of the possibility that some of the drug resistance issues could have started anywhere and gone in any direction. So I'm delighted to see Akhil is here. We have a fantastic scientific advisory group that has visited our sites. This is at Goa Medical College. This is Dr. Edwin, Dr. Edwin Gomes, who is the head of the Department of Medicine. And he's been terrific about working very closely with us. Goa Medical College gave us space right above the medical wards, and so our lab is built immediately above where the patients are. You'll recognize a lot of these folks here. I particularly want to single out Alan McGill, who unfortunately passed away at the end of last year. He was a terrific supporter and very understanding of various issues we have to deal with. These are our first hires. Anjali Mascarenhas had moved from the US back to India. She has a PhD in biochemistry from University of Illinois and did a postdoc in Ohio State and wanted to be close to her family. And she's the anchor for our Goa group, which has grown quite a bit. There are clinical research assistants in here, there are molecular biologists, cell biologists. We've now even hired 2 postdocs from IIT Mumbai that all the other major corporations want to bring to the US. So we're beginning to live our dream of actually getting all the really high-end if not expertise, certainly potential into our program. So here is actually real data from our activities at Goa Medical College over 4 years. The lines here show the rainy seasons. The bars show the number of patients we get. The yellowish green bars you see are Vivax. The red bars are falciparum, and then there's some mixed infection. And what you'll notice is that there were very definite peaks up and down, and then it looks like it may start flattening out here a little bit. Something strange is going on. This is a 1-year data set. If this trend continues, we'll want to keep an eye on it. There's plenty of Vivax. People like Manoj who want to work on developing methodologies for Vivax, such as cell culture system, love the fact that they can get 7 fresh Vivax samples per day in the middle of the malaria season. And it's really become incredible for studying Vivax. We also, of course, get more than enough falciparum. With all the things we do with culturing falciparum and gametocyte production and all the other activities, we get more than enough falciparum. And I'll show you the real numbers. But the other interesting thing about Goa is because it's one of the richest states in India, small as it is, it draws people from the poorer states to come work as sort of guest workers. And they're often involved in the construction industry, but they're also involved in the fishing industry and so on. 60 to 70% of the patients that we see in Goa are actually coming from other parts of India. So this little heat map is actually telling you, based on interviews, where they came from in the last month, where have they traveled in the last month. And you can see that sitting in Goa, we may actually be capturing cases of malaria coming from other parts of India. If nothing else, it's a really good training set of samples for us to learn how to deal with diverse parasite samples. Then we can go to the sites in the Northeast and compare what we learned from Goa with these samples. So I thought I'd throw out some numbers. During the period that this data that I'm showing you was collected at Goa Medical College, this little hospital with about 400 beds saw about 1.5 million patients that are coming through as outpatient. There are about 200,000 inpatients. I'm told there are about 74,000 febrile cases and about 6,000 cases of malaria. Quick judgments are being made on whether they should be admitted or not. And often it's based on clinical diagnosis that I'm sure is very accurate and valuable, but not very well spelled out. And often it's young residents that are making decisions at odd hours and so on. But we collect and capture as much information as we can. The other thing I wanted to show you was our own data in terms of how many patients we see in every season and the fact that they're all going up. If I were to show you the data which is publicly available from the government of India for this particular state, not one hospital, but the whole state, it's a small fraction of what we're seeing here. So again, it validates the fact that there is need for more accurate data all around India in terms of how much malaria there is and what kind of malaria there is. We take some of these data, and as Mala was saying, put it into machine learning systems that help us prioritize what might be good indicators for people who are working in a low resource setting and have quick decisions to make. And there's a paper coming out soon where we actually recommend that what are the things worth paying attention to and what may not be worth paying attention to in terms of deciding who is at greatest risk for getting very sick. And we don't necessarily understand all of that. In terms of basic science, we're really interested in phenotypes. Joe Smith, Manoj, myself, we all make a living by studying parasites in culture. But now we have the luxury of having history of these parasites because of what we know from the patient, where they came from, how they responded to treatment in the hospital, and so on. We learned that even just adapting parasites in culture is not trivial. And our staff had to be trained locally. This is particularly true because there are a lot of restrictions on how much samples can move around. We don't have the luxury of bringing all our samples to Seattle and working them up. We do everything in Goa. Almost everything I'm going to show you was actually done in Goa. So John White, who some of you know well, really did train people very well. We did some very systematic studies to look at different patient samples shown here. How long did they take to adapt in culture? Some of them would adapt as quickly as DD2 coming out of the freezer. If you know, DD2 is one of the most adaptable parasites we all work with in the lab. Others would take up to 2 months before they would adapt, and some would never adapt. So we would see these large variations. Of course, patients are coming in at different times of the month. They're being exposed to different media and different red cells. But what we learned was that if we go back to the freezer of parasites that have been saved, and we go back and thaw them out and put them in the same media with the same red blood cells and ask how quickly do they adapt to culture, they roughly follow the same trends. I can go into why the two don't, but most of them do. The ones that adapted fast, straight from the arm, adapt well even a couple of years later. Culture adaptation is one of the first major challenges these parasites face when they go into laboratory conditions. And we keep track of these parasites, both in terms of whether they were the fast adapting ones or the slower ones, when we start doing other things with them, including challenging them with antimalarials to see if they will rapidly change. I just wanted to touch on a few different things that we've learned that we could never learn in a laboratory, say in a Western country. These are things you can only learn when you work straight with patients. When we arrived in India, the first recommended drug cocktail that was being used in India was a combination of artesunate, sulfadoxine, pyrimethamine. And we were a little bit surprised because there had already been papers for about 5 years showing that different parts of India had high-level sulfadoxine pyrimethamine resistance. But the feeling was that in clinical trials, the combinations of pyrimethamine, sulfadoxine, plus artesunate were working well. Anyway, we went ahead and collected a whole bunch of different parasite samples shown here in a coded form, did genotyping on them. And they had maybe 2-point mutations in DHFR, sometimes one more in DHPS. This would predict sort of moderate level pyrimethamine sulfadoxine resistance. When we do the assays in the laboratory with normal media, which we all use in the lab, all of them were as resistant to pyrimethamine and sulfadoxine. I'm just showing you the sulfadoxine data here. They were as resistant as DD2 in media. So this is these are log scales, by the way. So compared to a highly sensitive strain, every one of these parasites that we looked at was about 10,000-fold resistant to sulfadoxine. We then looked at EC50s in the presence of not minimal media, but media without high levels of folates. And it's been known in the field, starting with Will Milhouse, and then later on John Hyde did a lot of work to show that parasites can use folates from the media and become resistant to antifolates. And the phenotype is not fully understood. We actually don't know what the mechanism is for this folate-dependent resistance. But clearly that plays a role here, because if we remove folates, then they become slightly more sensitive, but they're still 100-fold more resistant than highly sensitive lines. And then what was really surprising is there are some lines that don't care whether they're folates or not. So these parasites are, again, 10,000-fold resistant. They don't have the classical high-level DHFR sulfadoxine-resistant mutations, and we clearly don't know what the basis for this is. We've done whole genome sequences on these parasites, and it's not the classic things everybody talks about: no amplification in GTP cyclohydrolase, no mutations in the folate transporters. We don't know what it is. We're going to work with Stefan Cappe and Ashley, who have a humanized mouse model, to do some genetic crosses and find out what it is. I'm trying to capture this idea of going into the field, knowing a lot of details about basic science, and then asking questions like this. So the paper that's flying around, I'm so proud of this title, but I think it's putting people off, which is False Security from Obsolete Malaria Drug Resistance Markers. There is a whole industry of people who go around sequencing DHFRs and SP-resistant markers and making clinical judgments on whether these particular drugs should be used in a particular place or not. It's not so clear that that's always helpful. Clearly, clinical studies are very helpful, but they're expensive and difficult to do. But cell-based assays are also important. And so you need this continuum all the way, not in one central place in Delhi, but in many other places. Just a reminder to myself to stop talking. Similarly, for the pathogenesis studies, we have questions related to cytoadherence using cells that come straight out of the patient. We have questions related to invasion pathways, because a lot of what we know about invasion pathways comes from cultured cell lines. And Manoj and his group are very interested in what happens in a natural situation, different from what has been studied a lot in Africa. And then, of course, there are a lot of questions waiting to be answered with Vivax if we can get our culture systems in order. And I think short-term cultures are working very well, where we can look at invasion and growth. And you'll probably hear about it from Manoj soon. One of the great stories that came out of these types of comparisons, the clinical history of the patients, the degrees of disease severity, the type of parasites that came out of these different patients, their ability to bind to EPCR receptors. When you combine all of these together and put them again into the types of methods that Mala has been encouraging us to do, which is machine learning, and feed all of these different parameters, certain things jump out. And in this case, it turns out that there's one particular VAR gene family, which has DC8 and DC13 domains, which in Africa had been shown to interact with EPCR receptor on endothelial cells of capillaries from brain tissue. But also only the whole thing becomes really relevant when you combine it with measurements of high parasite biomass. So it's the combination of high biomass and the expression of these particular type of VAR genes that is actually a very good predictor of how sick people will get. And because we have the collection of clinical data, the RNA sample saved, and we have parasites available in culture that can be selected against the brain endothelial cells, that you can combine all of this information and come up with some things along these lines. There's a lot more work to be done. This is now when I start, because now I see the interactions I care about. I want to know how much this changes, how many mutations occur as you run into different types of humans with different types of EPCRs and so on. It's because it is ultimately about how quickly the parasite changes as you get into these issues. Invasion, Manoj and his team are doing some great work looking at ligands and alternate pathways of invasion that hasn't been studied well enough in the settings that we're in. We also have really nice collaborations coming in, such as Julian Rayner from Sanger, who's got some nice antibodies that he predicts are going to be of general use, and they're behaving very well with our Indian line. So these antibodies are blocking entry of parasites into red cells. For us, again, it's the beginning. Can we actually select for parasites that overcome these antibodies? Immunologists have not asked enough of these questions. We want to do what we do with drugs, with potential vaccines. And our vaccinology friends are courageous enough to let us try it, and it's great fun. I want to touch a little bit on integrating information from hospitals with things that go on in mosquitoes. And as I mentioned, Dr. Ashwani Kumar from NIMR Goa is down the road from the medical school. He's hired a lot of people from the Isomer program as well as his own Indian government resources. I want to single out Praveen here. Praveen actually spent a lot of time in the Mid-Atlantic area. He was a graduate student with Akhil, and later on he actually worked at NIH as a postdoc before he went to India, and we're really fortunate to have people like him working with us. He worked with Ashwani Kumar's group, and they have been collecting mosquitoes where we see a lot of malaria. Construction sites where people are coming from northeast parts of India, and they end up being sick. They come to Goa Medical College because that gives free health care to all of them with no questions asked. And again, there were prevailing ideas of what are the major vectors that carry malaria in India. Those of us who have been working on combinatorial chemistry to develop drugs or looking at genomics to look at which genes are important have abandoned the idea that we know things. We try to capture as much information as we can. And in that spirit, we all agreed that we should actually go out and collect mosquitoes and see how much of them carry human malaria. And out of that activity, they collected about 23,000 mosquitoes that included about almost 1,400 anophelins, and then they tested them for whether they carry human malaria parasites or not. And I'll give you the punchline. Ignore the spots on the top. Those are coded things about whether they have vivax or falciparum or mixed infections and so on. What's important is that Anopheles stephensii, which is considered the major urban vector in India, peaks right after the season here in the blue line. And what we found is that there is another vector called Anopheles subpictus. And Anopheles subpictus actually peaks alongside with Anopheles stephensii, and it actually carries human malaria parasites just like stephensii. What's even more interesting is that it comes up during the dry season. It turns out that it can grow fairly well in salt water, and it has this second transmission peak where stephensii is not carrying malaria. And we think that this may be one of those types of things that explains why you get year-round patients showing up in our hospital because there is year-round transmission going on. Sometimes it overlaps with Stephen's-I. Other times it's this new vector that's carrying malaria parasites in our neighborhood. There may be many such questions. This has captured the attention of the National Vector Disease Control Program. They want to carry out such analysis in other parts of India, which have many other different variant types of vectors. We want to test some of these ideas back in the lab. India has a huge 100-year history of growing mosquitoes in the lab, but not feeding experiments. And so we learned to do our own feeding experiments. Again, Praveen and Ajit were very involved in this. And we now can get infections in the gut. We get parasites, sporozoites in the salivary glands. And we're actually working with Jetsuman and with Rapat in Thailand to actually develop a liver infection system too. So all of these together, I think, will increase our capacity for preparing for what is going to happen. All of this is also forcing us to start thinking about how we're going to tell what parasite we're working with. Patient history is fine, but we want to be able to combine that with genotypes of parasites. And there have been a lot of, maybe the next slide will be more useful. Folks like Dominique at the Sanger Center have shown you plots like this, where you look at whole genome sequences of parasites and you line them up on their relationships. And what you find is that they cluster based on continents. And India was always missing. And we've taken our parasites from Goa, and what we find is that they occupy a very distinct space in this architecture of genomes. It's somewhere between what you see in Bangladesh and what you see in Myanmar. And there are some assumptions about where these parasites come from. We're very anxious to go and now add more parasites from our different collection sites in India and see if these branches continue to go out or whether we get new branches. The message here is that India has a very distinct genotype of parasites. It reflects the nature of humanity, the vectors, the vegetation, and everything that allows those parasites to flourish in that environment. I'm going to quickly jump through some things that are going on in the Northeast. We'd never done a community survey. Our scientific advisory group encouraged us to do this. We have a site in Northeast India that has about 7 villages, and Laura and her team have actually worked very hard with the local healthcare workers. These are very remote parts of India and some of the most poor parts of India, but have a lot of malaria. And again, as it happens in many other cases we've heard here before, some of these are also politically very unstable places. And we have good relationships that allow us to go in those sites. The community workers visit individual homes. They collect blood samples regularly in an active surveillance program. These are not easy places to go to. This is why if we see interesting cases of drug resistance in these areas, there are good reasons to believe that they originated there. And we have the genomic tools now to ask questions like that. And that's all going on. We have one more year of the isomer left. I'll skip this slide and basically show you that in some of these villages, as many as 20% of the people were positive for malaria when we did an active surveillance program. And many of them repeatedly get malaria even after we've left during the active surveillance. And we record febrile cases that are positive. Now we've started culture systems in Northeast India, so all the things we're doing in Goa can now be repeated with parasites from Northeast India. And we're doing many of the things that others are doing, where we combine information from hospitals with in vitro cell culture assays, expanding our assays for what is coming next. Drugs that attack not just the blood stages, but other stages of infection. We should be ready and prepared to be able to assay for potential resistance against that. And I think I should stop here and wrap this up. There are a lot of people to thank. I've tried to mention some as we go along. And I'll stop here and answer questions.
We have time for 1 or 2 questions. I think might be off the hook.
There's the time to go to the mic.
Oh.
Hi, I'm very curious. I know you didn't have a lot of sequencing data from the Indian samples yet, but were any of those from the section that's on the other side of Bangladesh? Was there any?
So, yeah, as I pointed out, the sequences on which the chart was drawn were samples collected in Goa. There are good reasons to think that some of them come from the other side. But we now actually have sequences currently running within India that are samples collected directly in the Northeast, including from the community survey. So we're very excited about what's going to happen in the next few weeks.
Thank you.
Thank you.